All right. Hello, everyone. Welcome to the second Rising Star webinar. My name is Yuing Chen. I'm a postdoctoral fellow at the National Institute of Environmental Health Sciences of the United States. As a member of the Virtual Education Committee of SSR, I have the honor to moderate today's webinar with two amazing speakers, Dr. Tessa Lord and Dr. Kate Greif. Each speaker will present for about 20 minutes, followed by a 10-minute Q&A session. Please type your questions into the Q&A box and I'll be moderating those. Our first speaker is Dr. Tessa Lord. Dr. Lord is a senior lecturer in biological sciences at the School of Environmental and Life Sciences, University of Newcastle in Australia. Today, she will be sharing the phosphoproteomic analyses identifying novel regulators of spermatogonial stem cell function. Let's welcome Dr. Lord.
Thank you. I'll just share my screen. Does that all look okay? I'll assume
Yes.
that it does. Thank you. All right. Thank you, Yuying, for the introduction. And I just wanted to start by thanking the organizers for selecting me for this SSR Rising Star accolade. I'm very humbled and appreciative for the award. So thank you for that. As Yuying said, today I'm going to be telling you a little bit about a phosphoproteomic database that we produced using mouse spermatogonial stem cells. And I'll tell you a little bit about some of the potential master regulators that we've identified from this database, and some of the validation kind of follow-up studies that we've done there. So this is really a project that's been born from a collaboration between myself, a spermatogonia biologist, and Dr. David Skerritt-Byrne, who's now at Helmholtz in Germany, who is really a proteomics expert. So I'm sure that this audience doesn't need too much of an introduction on the topic, but I just wanted to refresh everyone's memory that the spermatogonial stem cells, or SSCs, are of course the stem cells in the testis. And these are really the cornerstone of ongoing male fertility. So the stem cells in the testis, like in any tissue type, have the capacity for self-renewal to maintain that reservoir of spermatogenesis. But they also can give rise to progenitor spermatogonia that are essentially poised to go through differentiation upon receiving the retinoic acid signal that's produced by the somatic cells in the testis. Some key points of context, I guess, for my talk today is that the SSCs are defined in the testis around the time of birth, but this process of producing mature sperm won't happen until puberty. Also important to note is that the SSCs are a really rare population of cells in the testis. So today I'm going to be focusing on the SSCs and the progenitor spermatogonia, which are considered the undifferentiated population. And I'll also be talking a little bit about the differentiating transition as well. But I kind of wanted to touch on why we might want to have a better understanding of kind of master regulators of fate decisions in these spermatogonial subpopulations. And my research group is particularly interested in technologies that might use SSCs. And the one that probably comes to mind, first of all, for everybody, are these kind of pipelines that are in development to try and reverse infertility in survivors of childhood cancer. And this is just one simplified example. There are multiple pipelines being developed. But for simplicity's sake, we have a prepubertal patient that's not yet making sperm, so they can't cryopreserve a semen sample to safeguard their future fertility. But what we may be able to do instead, and certainly what's already being offered at clinics around the world, is cryopreserve a testis biopsy that contains SSCs. And the hope is that when this patient recovers and wants to start a family, that you might be able to expand those stem cells in culture and transplant them back into the patient's testis to restore spermatogenesis and hopefully his natural fertility. An SSC technology that you may be less familiar with that we are also very interested in here is the idea that you can adapt these kind of pipelines in the wildlife conservation space to try and promote genetic diversity in vulnerable or endangered species. The main difference in the pipeline being that now you have a donor and recipient animal that are different animals, and the recipients may be of the same or a closely related species. And in my lab group, I have a PhD student, Kat, who is working on a project surrounding this, particularly using koalas that are endangered in our home state of New South Wales, and that also produce sperm that don't survive cryopreservation. So if this is a topic that interests you, Kat and I have published a review on this in BOR last year, so I'd encourage you to have a look at that. But whether we're talking about either of these pipelines or things like in vitro spermatogenesis, in all of these cases, we really need to have a better understanding of factors that regulate SSC self-renewal and their differentiation, so we can really harness these in vitro technologies. So I mentioned phosphoproteomics. Why might we want to do phosphoproteomics? For those of you in the male germline stem cell field, or even in any field at the moment, you'll know that there has been an explosion of transcriptomic databases that are out in the world now. So with the advent of single cell RNA sequencing, and I did a quick PubMed search, and I found at least 60 that capture our SSC and progenitor populations that we're interested in, including some that I've produced myself. So there's a lot of information out there now about these cell types that we're interested in. But we were thinking that, there are still limitations to these data sets. And the main one that we were considering is that transcript abundance isn't necessarily equivalent to protein abundance within the cell, and certainly not protein function. And in fact, there was this paper that was published in Cell in 2020, where they showed that in the testis, more than 50% of genes actually have discordant expression, which means that the levels of transcript and protein don't actually correlate. So what we wanted to do is complement our transcriptomic data with a proteome and phosphoproteome for our populations of interest, so we can have a better idea about the presence and abundance of the proteins, and also their activity or their signaling and function within the cells. I make this sound like this is a really easy thing. It's not. Obviously, historically, there's been a lot of roadblocks to doing this. One of them for our cell type specifically is that it has been difficult to isolate pure populations of SSCs and progenitors. But really the main one for everybody has been that for an in-depth proteomics database to be produced, you really needed large numbers of cells or a large concentration of protein, which is something that's very difficult when you're working with rare cell types. So I'll walk you through this pipeline that we developed to overcome these roadblocks. And I guess the first really important tool is that we were using a transgenic reporter mouse line. So this was the ID4 EGFP mouse that was produced by John Oatley's group at Washington State University, where I did my postdoc. And there's been a lot of publications out there in the literature characterizing this mouse and the different populations. So I'm not going to go into this in a lot of depth. But essentially, this is a tool that we can use to isolate out the SSC and progenitor populations that we're interested in. And this is based on the intensity of their GFP fluorescence. So basically, it's been shown that the cells with the bright GFP fluorescence are a relatively pure population of stem cells, whereas those with dim GFP fluorescence are the progenitor spermatogonia. The other key component of our pipeline was this EasyPhos platform that was developed by Sean Humphrey at the University of Sydney. And this has really revolutionized the ability to do phosphoproteomics with rare cell populations, because you can get away with, I think, using as little as 20 micrograms of protein, which is a big jump on what was required previously. So we ran our cells through this pipeline. Again, we were looking at SSCs and progenitors, our undifferentiated spermatogonia. And I'm going to walk you through some of the data that we've produced, starting by looking at the proteome itself, and then I'll move on to the phosphoproteome part a little bit later. So firstly, we were happy that we produced a comprehensive data set. So you can see we identified more than 8,400 proteins in each of our populations with a good degree of coverage. And this is really a considerable jump beyond what was out there previously. So you can see in some cases we have more than 4 times the amount of proteins that have been identified in previous publications. But importantly, in these previous publications, these are all heterogeneous populations of spermatogonia. So in other words, the stem cells and progenitors are intermingled together. So they're not pure populations like in our database. And also, I think all of these previous studies expanded the cell population in cell culture beforehand. So doing this kind of in vitro expansion can also introduce some artifacts, I guess, into your data. So our cells are taken directly from the testis. In terms of proteins that were identified in our populations, you can see that there's quite a lot of overlap between the two. And this is really what we expected to see, because the SSCs and the progenitors are cell types that are on a continuum rather than being two completely distinct cell populations. So we do expect that the core proteome would be largely the same, and that's what we see here. But for the 8,443 proteins that they have in common, when you look at this volcano plot, you can see that actually over 500 of these have significantly different expression between the two populations. For instance, in the SSCs, more than 250 proteins have significantly increased expression. And I'll come back to what this tells us about these cell populations in a moment, but I just wanted to revisit quickly this idea of discordant expression between protein and transcript. We overlaid our proteomic database with a previously published RNA-seq database, and this was a database that was using exactly the same cell populations isolated from the same mouse line. And you can see that there is a relatively weak correlation between transcript and protein abundance. So again, just highlighting the need for caution when using transcript abundance as a proxy for protein abundance and activity within the cell. Okay, but we have this list of differentially expressed proteins between our stem cell and progenitor populations. We want to look at this and try and learn something new about these two different cell types, and also identify some potential, I guess, regulators or master regulators of their activity. So to start doing this, we put our list of differentially expressed proteins into Ingenuity Pathway Analysis, or IPA. And basically, this will give you a list of significantly enriched pathways, and it will also predict upstream regulators of your changes in protein expression. IPA, I should say, also gives directionality in terms of the readout, which is what the activation and inhibition symbols are on the heat maps here, but I'm not going to kind of touch on that too much today. Firstly, what we were happy to see in our IPA analysis was a number of pathways that have been previously characterized to be important for SSC and progenitor function. So for instance, we see RET signaling, which we know is very important for driving self-renewal in SSCs. We see mTOR signaling, which is important for progenitor spermatogonia. And then in our predicted upstream regulators, we have, again, some really well-known candidates here. So for instance, GDNF and FGF2 are growth factors that we know are produced by the Sertoli cells that act on the SSCs in a really kind of indispensable manner to drive their self-renewal. So essentially, the first thing that came out of this analysis was that we were reassured that we had produced a biologically relevant data set that reflects what we already know about these spermatogonia populations. But of course, what we're trying to do is identify some novel regulators, and we certainly were able to pull quite a few of those out of the data set as well. So I'm just going to focus on one of those now, which is this growth factor PDGF. So we wanted to further explore whether PDGF could be an upstream regulator of spermatogonia function. The first thing we thought we probably needed to do was make sure that our undifferentiated spermatogonia actually expressed the receptor for PDGF. If they didn't, then it wasn't really worth following this up any further. But you can see this is just a simple immunofluorescence experiment, again using the ID4-GFP transgene to highlight the undifferentiated spermatogonia. But you can see that they do indeed express the PDGF receptor A isoform. The next thing that we wanted to do is make sure that the PDGF ligands, of which there are A through to D isoforms, that they're actually expressed by some cell types in the testis. In other words, that this is a growth factor signal that the spermatogonia would normally be exposed to in the testis. And this is just the results for the B isoform here, but you can see that there is strong expression. There's expression across all A through to D. What we tend to see is the highest level of expression in the somatic cells, so the Sertoli cells and the Leydig cells, but the expression tends to be excluded from the spermatogonia themselves, which is more evident in this whole mount analysis here. So in other words, what we were thinking could be happening is that these PDGF growth factors, like with GDNF or FGF2, could be produced by the surrounding somatic cells in the testis and then acting on the spermatogonia in the niche to instigate some sort of, intrinsic signaling pathway and some sort of cell fate decision. So of course we wanted to know, what PDGF is actually doing to the spermatogonia. So we've initiated a pilot study using undifferentiated spermatogonia in primary culture in vitro. So we're supplementing with some different PDGF ligands here. And I should point out that these cultures also carry this ID4 GFP transgene, so we can monitor the percentage of GFP expressing cells as a proxy for the number of stem cells within the culture. And essentially what we can see is happening over 4 weeks is that the cultures continue to proliferate with PDGF supplementation, but the number of stem cells seems to plateau. So we think that this PDGF growth factor that was identified in our proteomic data set may actually be a growth factor that stimulates the SSC to progenitor transition rather than SSC self-renewal. And this is something that we're continuing to investigate. So to jump back now to talk about the phosphoproteomic results, as far as we know, this is the first phosphoproteome that's been produced for our SSC and progenitor populations. And again, we were happy with the depth of the data that we'd produced here. So you can see more than 3,700 phosphosites were identified. This time we see quite, I guess, an interesting shape to the data set, and you can probably appreciate this most by looking at the volcano plot. So what we saw was kind of a wave of hyperphosphorylation upon the progenitor transition. And we wondered whether this was reflective of kind of a flurry of activity in these progenitors that was essentially preparing them for differentiation. In terms of pathways that are predicted to be regulated by these differential phosphorylation events, we see things like histone modification, repair of DNA damage, the cell cycle, pluripotency, and apoptosis. But what we wanted to do is try and, again, perform some functional analyses to validate this. And we wanted to try and predict some master kinases that might actually be driving this wave of phosphorylation and inhibit them in our cells and see if any of these processes were affected. So David has an in-house pipeline, I guess, to try and identify master kinases based on the data that we produce. He's published it previously in this paper at the bottom here if you're interested. But essentially it combines the IPA software with something else called Phosphocyte Plus, and also kinases that are expressed in our proteomic data set. So essentially we narrowed it down to 3 candidate master kinases that we knew inhibitors already existed for. And we elected to follow these up further by basically inhibiting them in our primary cultures of undifferentiated spermatogonia for 24 hours and then assessing what the response was. So the first thing that we saw is that there was no real change in the proportion of SSCs and progenitors in these cell populations in culture, at least within the time frame that we looked at, with our kinase inhibitors. And I should point out here that the trio bar at the end is basically all 3 being inhibited at the same time. So no change there, but what we did see is that when we added retinoic acid to the culture well to stimulate the differentiating transition, that there was a significant reduction in the number of progenitors that were able to differentiate as assessed by c-KIT expression. And there's still additional replicates of this coming along now. So again, what we think might be happening is that these master kinases are preparing the progenitors for the differentiating transition rather than driving the SSC to progenitor transition specifically. The other thing that we saw was a significant reduction in overall cell number when we inhibited these kinases. We thought that this could have something to do with reduced proliferation, but we didn't see any real changes in the cell cycle. But what we did see, and these are still, again, ongoing experiments, but evidence for increased apoptosis. So an increased percentage of tunnel positive cells and increased caspase-3 activity as well. So we think the other role for these kinases may also be a protective kind of function. So to start to wrap things up, we produced a comprehensive phosphoproteomic database for SSCs and progenitors. And we're hoping to provide this as a kind of resource for the field. So we're producing a Shiny app that will capture both the proteome, the phosphoproteome, the master kinase list, and the overlay of the proteomic and transcriptomic data. So everyone will be able to access that. As I said, we identified key signatures of our spermatogonia subpopulations, however, also some unique insights above that of previously published transcriptomic data sets. An example of a potentially novel regulator that we've identified is this PDGF growth factor that I think might be kind of supporting the SSC to progenitor transition. And then with our phospho data set, we've also been able to identify some potential master kinases, and we're performing some functional analyses. So again, we think these that we've identified might actually be preparing the progenitor spermatogonia for the differentiating transition. So essentially, we hope that with improved understanding of how fate decisions in spermatogonia are regulated, we can continue to develop these kind of in vitro platforms, for instance, for reversing infertility in survivors of childhood cancers, or in that wildlife conservation space that I was talking about. So with that, I'd just like to thank a few people. Firstly, I'd like to thank David, without whom this wouldn't have been possible. I'd also like to thank the members of my group, our funding bodies, and a special shout out to Neve Ford, who actually nominated me for this Rising Star Award. Thank you for being my cheerleader, Neve. And I'm happy to take any questions. Thank you.
Thank you for a really fascinating talk, Tessa. We already have one question in the QA box. Fantastic work, Tessa. I imagine this is a difficult question to get at, given some of the limitations of the ID4 EGFP mouse, but would you expect to see proteomic differences between neonatal versus adult SSCs?
Yeah, thank you, Tegan, for the question. Yes, I would expect to see differences. As you mentioned, there are limitations just because we have to add, I guess, additional fluorophores into the mix to isolate out the stem cells from the adult testis. And because they're, even more rare at that time frame, it becomes quite challenging. But previously, we tried to kind of answer this question, I guess, by comparing in our RNAi screen, comparing cultures that had been initiated from the postnatal testis versus adult testis, the stem cells that we'd isolated from there. And there were definitely differences in how they respond to knocking down different transcription factors that we think are master regulators there. So I do think that there would be differences. It would be a great thing to follow up, but I think even with the progress that we've made in the proteomics pipeline that we're using, it would still be very challenging to do this with the adult cells.
We'll have time for a couple more questions if people want to put their questions into the QA box. So I guess I'll ask one. I am wondering if it's possible to estimate if the discrepancy that you're seeing between the RNA versus the protein data is because that they're not being translated versus it's the sensitivity of like your proteomic detection method.
Yeah, it's a great question. And I guess I'll refer back to, so talked about the Cell paper that was published in 2020, where they showed discordant expression between essentially every tissue type. But the testis was really kind of a leader in that the testis has the most enrichment for RNA species. So in other words, the RNA tends to be more abundant than the protein. So I think it's that there's a lot of RNA that's not being transcribed to protein. So often if you're using that as a kind of readout for protein activity in the cell, it may not be an accurate reflection.
Thank you. So we have a second question coming in. Are the master regulators regulated?
Thank you, Kate. Yes, I imagine that they are. And so this opens Pandora's box, doesn't it? And we haven't started to explore what is upstream of those. So that's definitely something that we could follow up once we're, very secure in the fact that they are having a key effect when we inhibit them in the spermatogonia.
Someone asked how it's different from single-cell RNA-seq data with proteomic data.
Yeah, so we haven't overlaid our proteomic data with the single cell readout specifically. The reason that we picked the bulk RNA-seq to do the overlay is because we knew that these were produced from exactly the same cell populations from the same mouse line. And so I guess you'd just be introducing some more variability by overlaying it with a different single cell database. But I would still, expect that there would be that discordant expression that I mentioned, but perhaps in a less reliable kind of readout than what we'd see with the bulk comparison.
Next question. Great work. I understand that the study can contribute in reducing infertility in men. However, my question lies in the fact of how much time this whole process can be established for detecting infertility.
Yeah, so I guess, so as I mentioned, we're particularly interested in developing kind of in vitro pipelines to address very specific types of infertility. So in that the childhood cancer kind of space, you would be potentially expanding stem cells in a culture dish and then transplanting them back into the testis that is devoid of germ cells so they can repopulate and reinitiate spermatogenesis. I guess for other types of infertility in, a perfect world, you could also try and detect, what signaling pathways have gone awry and kind of target them with specific molecules. But, that's going to be a difficult thing and not something we've really considered at this point in time. But yeah, what we're really trying to do is improve our capacity to target and kind of drive specific fate decisions in the SSCs in vitro for those kind of technologies and platforms to address certain types of infertility.
We have one more question in the chat. Excellent presentation, Tessa. Do you intend to assess the evolutionary conservation of the differentially expressed proteins and their expression patterns in non-model species during spermatogenesis?
I would love to do that. As I mentioned, we have a particularly keen interest in the koala. So we, at the moment, are particularly interested in what's conserved between our mouse models and the koala. But we do also have access to a lot of other cool species here in Australia. So yes, it's something that I'd love to do if we had the funding to do it.
Thank you so much, Tessa. So we'll move on to the second speaker, but if you have more questions, please direct them to Dr. Lord. Our second speaker is, thank you. Our second speaker is Dr. Kate Greif. Dr. Greif is an assistant professor of obstetrics and gynecology from the Laboratory of Reproductive Research at Women and Infants Hospital of Brown University. She will be discussing the diverse roles of the proteostasis regulator, ubiquitin C-terminal hydrolase L1 in female reproductive health. Dr. Greif, the floor is yours.
Thank you so much. Let me just share my screen. Can we see that okay?
Yes.
Okay, Thank you. Thank you, Ying, for the introduction. Thank you so much to SSR and the organizers for inviting me here today. This is really an honor, and I'm really excited to tell you about some of our work on how oocyte intrinsic processes can not only affect oocyte quality, but also potentially overall reproductive and systemic health, as well as reproductive aging. So my slide would advance. There we go. So at its most simplistic, protein homeostasis or proteostasis requires the fine balance of three separate intricate processes. This involves protein translation or synthesis, proper folding of those synthesized proteins, and then degradation of old, misfolded, or excess proteins. That's important to dispose of them, but also to recycle their components. And this is via proteasomal degradation or other forms of protein degradation. And oocytes must perform this proteome regulation with high fidelity because they go through many complex developmental processes. And this includes germline cyst formation, which is typically in utero for mammals, followed by primordial follicle formation, or what we also know as ovarian reserve establishment for some of those oocytes, or a massive wave of fetal oocyte attrition for others. And after that ovarian reserve is established, oocytes will then progress through oocyte maturation and folliculogenesis. And it's during these processes that oocytes will undergo very active periods of growth, long periods of quiescence. Proteome regulation will also help with the maternal loading of organelles and essential proteins for embryogenesis and exclusion of protein aggregates. And as we know, oocytes are a finite, non-renewable resource. So getting it right the first time is really critical. And so how do oocytes regulate their proteomes, which must remain stable with high fidelity for long periods of time? Two recent studies have begun to shed some light on this, including work from Dr. Francesca Duncan and colleagues, as well as Dr. Melina Shu. And these studies are really seminal in demonstrating that oocyte and ovarian peptides are actually extraordinarily long-lived, with many of these proteins falling into really critical pathways and categories, which include regulation of the cytoskeleton, metabolism, mitochondrial function, response to stress, et cetera. And we're really interested in understanding not only how this process is accomplished, but also how accomplishing this process of oocyte proteostasis contributes not only to the health of the ovary, but also the systemic health of the organism. So how did we even get interested in this topic of oocyte proteostasis? We set out to ask a different basic question, which is, are there distinct molecular features of oocytes which will ultimately comprise the ovarian reserve? Which is to say, are there unique features of oocytes in a cyst which might predispose them to continuing towards a primordial follicle fate versus an oocyte attrition fate? So obviously a very different developmental question. And for a variety of reasons, COVID delays among them, we didn't end up actually achieving these initial goals as originally intended, but we did end up finding something that we found very exciting and that I'll be telling you about today. So to gain an understanding of early oocyte development, or the development of that early ovarian reserve, we profiled all ovarian cells from mouse ovaries at 4 perinatal time points. So this is embryonic day 18.5 to postnatal day 5, which is the time frame during which the ovarian reserve is forming in the mouse. And we actually had quite poor retention of oocytes in our samples, but we were able to identify oocytes based on known markers, and then looked at the top 20 most differentially enriched transcripts in that population, which you see in that heat map there. Many we know as master regulators of oogenesis or meiosis, but then we saw one, the 20th highest, the one at the bottom there, UCHL1, which was unknown to us. And so we chose to look into it further, as it had been previously known to be important in spermatogonial stem cell biology, which Tessa just beautifully introduced, including roles in regulating their self-renewal and metabolism. And as you can see from this t-SNE plot, UCHL1 is highly expressed in just a subset of the oocytes identified in this data set. So what does it do, or what is it known to do? It's classically a deubiquitinating enzyme, but it actually likely plays greater roles in ubiquitin homeostasis because its active site is quite small, and that precludes the binding of full-length folded ubiquitinated substrates. So instead, it likely stabilizes cellular monoubiquitin, hydrolyzes ubiquitin proproteins to recycle monoubiquitin, and or cleaves monoubiquitin from small disordered peptides or some degradation products. It may also have some non-canonical roles as an E3 ligase, which is in effect performing the opposite function. So it does seem that its function is very context and cell and tissue dependent. What we do know about it is best understood from studies in the brain, where it represents a staggering 1 to 5% of neuronal protein. It's essential for axonal health. It is known to be secreted by neurons. Through genetic manipulation or inhibition, we know that this can result in 30% less free monoubiquitin in the brains of animals that are treated in this way. We also know that using small molecule inhibitors in oocytes, that also reduces the pool of monoubiquitin. We have been fortunate to have access to global null animals, which are also known as gracile axonal dystrophy or GAD mice. These are complete loss of function animals, which unfortunately, because this depletes UCHL1 from the brain as well, means that they experience motor ataxia starting around 4 months and death by about 7 months. But they've been very useful for our studies, and I'll talk about them in a moment. UCHL1 is most highly expressed in the brain, pituitary gland, and the gonads. And this is all especially relevant as these organs comprise the hypothalamic pituitary gonadal axis, or HPG axis. And I'll get into that a bit more later. But in brief, the hypothalamus produces gonadotropin releasing hormone, or GnRH, which my cursor works here. And that stimulates the pituitary gland to produce follicle stimulating hormone and luteinizing hormone. And then the ovaries can respond to these gonadotropins by producing steroid hormones, estrogen and progesterone. And then these can all feed back to the relevant other organs to continue to regulate the cycle. And finally, there are a few known reproductive roles for UCHL1, and that includes aiding in the block 2 polyspermy. So we know loss in oocytes, again, either genetically or through small molecule inhibition, can allow for the binding and fertilization of multiple sperm. And there are known roles in SSC proliferation and metabolism, as well as progression of spermatogenesis. So first, we wanted to characterize protein expression of UCHL1 and to ask if it was in agreement with our single cell sequencing data. Again, as we just heard, these are not always congruent. So here what you're seeing are ovarian tissue sections from postnatal days 1 and 3 at the time of, or around the time of ovarian reserve establishment in the mouse, and 2 of the time points that we included in our single cell sequencing. So they're stained for an early oocyte marker, TRA9D8, which is on the far left column, UCHL1 in the middle, and then the merge shows DAPI in blue as well. And as you can see at postnatal day 1 on the top, a subset of TRA9D8 positive oocytes are also UCHL1 positive, but by just a few days after birth, virtually all oocytes have become UCHL1 positive. So we thought maybe this is actually that survival factor we had been looking for when we set out to do this initial study. We only, not only do we see UCHL1 in the perinatal ovary, but we actually continue to see high expression in all oocytes of all follicle stages. We do also see it eventually in the corpus luteum, or the remains of those ovulatory follicles, which produce progesterone after ovulation, but we do not see UCHL1 expression at the mRNA or protein level in any granulosa cells prior to that differentiation step. We also wanted to determine if UCHL1 expression possessed similar dynamics in developing human oocytes. So we obtained human fetal ovary specimens from our pediatric pathology department. So starting at 20 weeks of gestation and spanning to 36 weeks of gestation, we stained and quantified oocyte-specific UCHL1 expression in all present follicle stages. And we found that UCHL1 expression started to be detectable about 28 weeks of gestation, becoming highly expressed in oocytes of all stages by late term. And so in these images, the white arrows are pointing out just a few examples of oocytes in these sections. It's not exhaustive by any means. But you can see very limited UCHL1 expression in the 25-week oocytes in the top panel, but dramatically more in all the oocytes of the 36-week-old ovary in the bottom panel. And we also observed a stage-specific increase in UCHL1 expression when normalized for oocyte size. So this demonstrates increasing expression during human follicle development. So therefore, we do observe very similar expression dynamics in human oocytes to the dynamics we see in the mouse, and that gives us additional confidence that UCHL1 may be playing similar roles in human oocyte development and function, that we can use our mouse model as a good model for studying its function in the oocyte and the ovary. So that brings us to our original hypothesis, which is that UCHL1 was required for mammalian ovarian reserve establishment or maintenance. And to study this, we utilized or to test this hypothesis, we utilized that GAD global UCHL1 knockout mouse line that I mentioned before, and we comprehensively assessed folliculogenesis in these animals. And somewhat to our surprise, at 1 month of age, follicle composition in UCHL1 knockout animals, which are represented in red here, is grossly normal. And this is corroborated by normal anti-Müllerian hormone levels across genotypes at nearly 3 months of age. And that's sort of the gold standard ovarian reserve marker. Despite these quantitative findings, however, we did notice that we observed morphologically abnormal oocytes in our knockout ovaries, which are highlighted in those white arrows on the bottom right. And so we wanted to probe this a bit further because we thought that these initial data may not have been telling us the whole story. So to further this study, we then assessed fertility and fecundity in these animals, and we see that knockout animals, again in red, are severely subfertile. They produce far fewer litters overall and fewer pups per litter than their control litter mates. And again, interestingly, and somewhat to our surprise, heterozygous animals, which in here, in these bars and on these charts is represented in black, actually exhibit a bit of an intermediate phenotype. Important to mention is that we were only able to perform these breeding studies till 6 months of age, because it is after that point that, again, those global knockout mice succumb to severe neurodegenerative disease. So we're not able to profile their full reproductive lifespan. Additionally, when we stimulate with gonadotropins to do superovulation, we retrieve significantly fewer oocytes from knockout animals compared to control litter mates, suggesting that they are much less responsive to hormonal stimulation than their heterozygous or wild-type counterparts. So finally, when we look later on in life at 5 months of age, we actually observe that while the ovaries from UCHL1 knockout animals are significantly smaller than wild-type or het ovaries, as you can see in both the weight and the sort of macroscopic image, they actually contain greater numbers of immature follicles, which was a bit of a surprising result, but suggested to us that there may also be somatic cell dysfunction in these ovaries as well. And we wanted to probe that further. So given that we see those persistent follicles, but also reduced ovulatory capacity, we wanted to look at ovaries following ovulation induction. So as you can see on the top panel, following pregnant mare serum gonadotropin treatment, knockout follicles do not develop to the same antral stage as their wild-type counterparts. And then following administration of human chorionic gonadotropin, which is an ovulation trigger, in the bottom panel, you can see in the knockout case, many actually fail to ovulate. So as you can see in the wild type, we have these structures here. Those are the corpus luteum. And these are, again, the remains of the ovulatory follicles, which form following ovulation and will produce progesterone. And in the knockout, these structures form, but however, they prematurely luteinize without actual follicle rupture and therefore possess these retained oocytes, which are indicated here. And this is a bit of a unique phenotype, but there is some reports of it in the literature. It may actually be due to impaired gap junction formation or maintenance between the oocytes and their surrounding granulosa cells, loss of that communication between those compartments, and that may indeed lead to premature luteinization and failed ovulation. And we wanted to determine then if some of these phenotypes we were seeing were related to circulating gonadotropin levels, which are known to affect and regulate the functions of those gap junctions. So we analyzed hormones in the serum of 5-month-old mice. And indeed, we also see that knockout animals produce excessive levels of gonadotropins, which may render them unable to respond to additional stimuli and may contribute to that oligoovulatory phenotype. This was statistically significant for luteinizing hormone and was trending in that same direction for follicle stimulating hormone. Given the ovulatory failure or partial anovulatory phenotype that knockout animals possess and this clear dysregulation of the HPG axis in these animals, we wanted to look at granulosa cell health, which are those somatic cells surrounding an oocyte in the follicle. And as a reminder, these cells do not express UCHL1 at the mRNA or protein level until after ovulation when they're luteinized. So any effect on these cells, we presume, would be due to loss of UCHL1 from other cells or tissues. And as you can appreciate in these images, there's a qualitative increase in the number of follicles that possess tunnel-positive apoptotic granulosa cells in the knockout ovaries. And then when we quantify this, we see a statistically significant increase in tunnel-positive secondary follicles at 1 month of age, even in follicles which look otherwise grossly healthy. So to begin to explore why this might be happening, what potential mechanisms might be underlying granulosa cell dysfunction in these animals, especially considering that they themselves do not express UCHL1, we considered that essential role of bidirectional communication between the oocytes and the granulosa cells, which necessarily involves proper formation of the zona pellucida or the glycoprotein layer which surrounds oocytes. So we look specifically at zona pellucida protein 3 or ZP3, as this is one of the primary glycoproteins surrounding the oocyte, allowing for proper fertilization, as well as gap junction localization and to facilitate that communication with surrounding granulosa cells. And as you can see, ZP3 has a very different localization pattern in knockout oocytes, which are on the bottom panel. It appears thickened. In some cases, it's diffuse or wavy. And when we quantified the thickness of ZP3 staining in the oocytes of secondary follicles, and we did so over 4 quadrants for each oocyte, we do indeed see increased thickness, which in knockouts, which has also been associated with poor IVF outcomes in humans. And interestingly, may also contribute to that polyspermy phenotype that I previously discussed and had previously been reported in the literature in UCHL1 knockout animals. So as we observe phenotypes that suggest a loss of oocyte quality, we wanted to explore what UCHL1 may be doing in this context. And given its known roles in facilitating protein turnover, we approach this with quantitative proteomics of oocytes from wild type and global null animals. So what you're seeing in this heat map here are the top 20 most abundant proteins in wild type oocytes with the proportion of total oocyte protein that they represent and then compared to knockout oocytes. So reassuringly, we see a complete loss of UCHL1 from global null oocytes. And I want to draw your attention to the fact that this, in a wild type case, UCHL1 represents nearly 1% of all oocyte protein, which was quite shocking. But we see other interesting alterations as well. We see an increase in ZP3 expression, which corroborates our immunofluorescence findings. And we see a commensurate decrease in ZP2 abundance. Importantly, we do not see these changes at the mRNA level. So this is indeed suggesting it's an effect of synthesis or turnover of these proteins. And we know that the abundances of several hundred other proteins are also affected, including complete loss of many proteasome components, loss of some chaperones, and that just speaks to a very complex role that UCHL1 is playing in maintaining oocyte proteostasis. But as these oocytes are derived from global null animals, and we know that the HPG axis is generally dysfunctional in these animals, our next steps were to parse the effects of oocyte-expressed UCHL1 from other non-oocyte autonomous effects. So to start answering these questions, we first generated a floxed UCHL1 allele to allow for oocyte-specific ablation of the protein. I'll call these conditional knockouts from here on out. Indeed, when we look at the UCHL1 expression in these ovaries by immunofluorescence, we see complete loss of expression from oocytes, with the exception of a very few immature oocytes, and that's to be expected with the GDF9 CRE we're using. And when we look at isolated oocytes via Western blot, we see loss of UCHL1, but we don't see loss of the oocyte marker DDX4. But most importantly, perhaps, is we do not see loss in other tissues, including the brain. So in addition to testing the role of oocyte-expressed UCHL1, we'll also be able to evaluate the full reproductive lifespan of these animals without that hindrance of the neurodegeneration that we observe in our global knockout animals. So we're now working towards several goals. This includes quantitative proteomics of isolated oocytes and granulosa cells, quantitative analysis of differential ubiquitination of peptides in oocytes, which we hope to use to identify potential targets of UCHL1, evaluating regulators of bidirectional communication between the oocytes and the granulosa cells, as well as a reproductive aging study. And so to wrap up, I'm going to be showing you a glimpse into some of our preliminary and unpublished data that we've recently generated toward these goals. So first, we began by analyzing the zona pellucidas of oocytes from our conditional knockout animals. And what I hope you can appreciate is that in addition to sort of an increased density of staining of ZP3 in the zona pellucida of our conditional knockout secondary follicle oocytes, which is what we see in our global knockouts as well, we also observed that same thickening as well as perhaps sort of increased projections and artifacts. And that is something we observe in our global knockout as well. And so this leads us to believe that what we see in our zona pellucida phenotypes are indeed a result of oocyte-specific loss of UCHL1. And we're now working to quantitatively analyze these phenotypes, including thickness projections, as well as additional zona pellucida proteins. We've also begun to use our conditional model to explore non-cell autonomous roles of oocyte expressed UCHL1, first through RNA-seq analysis of wild type versus conditional knockout granulosa cells. And as you can see, and to our surprise, we see actually the vast majority of differentially expressed genes are significantly upregulated in conditional knockout granulosa cells, and that includes genes like OVGP1, which is up here. That's a oviductal glycoprotein 1, and it's expressed in the granulosa cells of preantral and antral follicles, but its role really isn't known, though there are some hints that it might be important for sperm binding. Increase in transcripts could be due to a number of different biological or artifact reasons. One potential explanation is ectopic expression of these transcripts in granulosa cells resulting from UCHL1 regulating transcriptional regulators or chromatin modifiers. It could be due to oocyte mRNAs inappropriately entering granulosa cells due to an impaired barrier between them. It could also be due to the inclusion of very small oocytes in this prep, as this is a prep of size exclusion. But even if that were the case, that would still be disproportionate in our knockout, as these were prepped the same between genotypes. And so it does still speak to some phenotype, and we're trying to resolve that now. What's also extremely exciting is we've also started exploring the role of oocyte expressed UCHL1 and its effect on the brain. So we performed a bulk RNA-seq analysis of wild type versus conditional knockout whole brain, and we actually find approximately 60 differentially expressed genes. Of note, many of these are related to extracellular matrix, intracellular transporters, immune-related transcripts, and we're validating many of these now. But this is all to say that it's really fascinating to us to observe how these changes occur in other cell types and tissues, given that this is from an oocyte-specific conditional knockout. And so we're really interested to explore the ways in which an oocyte-specific factor can influence both HPG axis function and then also this overall systemic health. And then finally, I want to touch on how does oocyte-specific UCHL1 affect these local and distant tissues? We hypothesize that oocyte-secreted UCHL1 can act in a paracrine and endocrine manner to affect reproductive function and overall health. As we know, it is also secreted from neurons. And we're currently undertaking studies to identify potential receptors, but I want to touch on another potential use for this. Serum UCHL1 levels historically have been used as a marker of traumatic brain injury, so increases in serum following these events, which disrupt the blood-brain barrier. But it is known to be a secreted protein, and its normal circulating levels under healthy conditions have just not been thoroughly explored. So based on our preliminary data from our lab showing much higher levels of serum UCHL1 in female mice and humans compared to males, we've now also looked at serum from our conditional mouse line, and we see much lower levels of UCHL1 in female conditional knockout serum compared to wild type serum, with an expected complete loss from our global knockouts. Suggesting that much of the serum level we observe in wild type females is actually oocyte derived and may therefore hold promise as a novel oocyte specific biomarker of oocyte quality or abundance. And we are now testing UCHL1 concentrations in follicular fluid in a pilot study of patients from our fertility center. And what you can see here is that UCHL1 is dynamic in these patients, and we are blinded to their particular fertility diagnoses for this pilot, but in some cases, it's 10-fold higher what we see in serum, suggesting a local source of secretion, which of course may influence the local follicle environment. And so we're now performing a prospective study to determine the association between UCHL1 serum and follicular fluid levels, and specific for infertility diagnoses, as well as its relationship to anti-Müllerian hormone and follicle-stimulating hormone levels. So while we still don't yet understand the exact molecular mechanisms for what UCHL1 is doing in the oocyte and ovary, we hypothesize that in the presence of UCHL1, ubiquitin molecules are hydrolyzed appropriately from protein degradation products. This allows for the restoration of the cellular pool. Ultimately, maintaining proteostasis allows for the maintenance of oocyte quality and architecture and healthy follicle development. And we do believe that UCHL1 plays some role in local paracrine signaling to granulosa cells, or potentially regulates the proteins which facilitate communication between these compartments, and may also play roles in more distant tissues as well. And on the other hand, in the absence of UCHL1, we hypothesize that ubiquitin is hydrolyzed less effectively, as has been demonstrated in the literature. This can lead to reduced cellular monoubiquitin, accumulation of proteins, and loss of oocyte quality. And indeed, we believe that the increased ZP3 expression, for instance, in UCHL1 null oocytes is a consequence of this mechanism. And with the loss of secreted UCHL1, we do believe we see altered paracrine signaling with surrounding granulosa cells. And that may also have farther reaching effects on distant tissues like the brain. So ultimately, we want to understand how UCHL1 regulates the oocyte proteome and proteostasis, because this may also shed a light on its role in human disease and ovarian aging. And with that, I want to thank my lab and all of my collaborators. The people bolded here on this list did the lion's share of the work, including some former fellows. I also want to acknowledge my funding, which has come from many sources for this project. And finally, for all of you for having me today and for your attention. And I'm happy to take any questions.
Thank you, Dr. Grai, for another fascinating presentation. If you have any questions, please put in the QA box. So I'll start with one. Do you know if the estrogen secretion is defected in either the GAD or the conditional knockout? And I'm asking, wondering whether the increased LH and FSH secretion is from the negative feedback of estrogen versus like pituitary cells are secreting secreting more because of UCHL1 defect.
That is an excellent question. So at the time we did this study, we were actually not able to measure it in our hands due to a discontinued assay. But a recent paper just came out, it was the end of 2024, with a different global knockout line. They did some of the same characterizations and some different ones, and they did indeed see reduced estradiol in those animals.
Okay. And okay, I don't see other questions yet. If UCHL1 is secreted by the oocyte, do you expect them to be picked up by the granulosa cells, like directly surrounding the oocyte? And if you would see them in staining in that case?
Yeah, we don't necessarily see it in staining. But we do hypothesize that it is acting locally on the granulosa cells, and certainly making it into circulation as well. I didn't have time to talk about it today, but there is evidence in other cell types that UCHL1 can bind EGFR and potentiate downstream signaling via EGFR. And so that's something we're actively testing right now in our granulosa cells.
Right. So I see a couple questions coming in. Does UCHL1 condition knockout have aging phenotypes, meaning loss of follicles over time or fertility issues similar to whole body knockout?
That's what we're actually doing that right now. And so our oldest animals are, I believe, only about 5 months old, which is basically where we had to, or close to where we had to cap our global knockout fertility analysis, because again, they have those neurodegenerative phenotypes. But we're trying to ask that very question right now. And we are very interested to know.
Okay, next question. Curious to know if you see any correlation between this protein with PCOS.
That is also a great question. So we thought about this as potentially a model of PCOS. And there are some similarities between the ovaries that we see in our global knockout animals and PCOS. For instance, we have this, increase, this persistence of immature follicles, partial and ovulatory phenotype. What we don't see, and what we did look at, was androgen levels. We don't see any increase in androgens. And so in some ways it models PCOS, and in some ways it doesn't. And, so there might be elements of PCOS that we could get at using this as a model, but certainly not all of them.
How do you think the ovarian factor impact the brain?
Also an excellent question. And honestly, we don't know. We are not certain at this time that UCHL1 can, bind to a receptor on a neuron and enter a neuron. And that's something that, one of the graduate students in my lab is going to be working on for her thesis project. But what we do know is that we have this panel of differentially expressed genes when our genetic manipulation is just knockout of UCHL1 from the oocyte. And so clearly it is doing something, and we're not sure if it's a direct effect or if it's some indirect effect of deregulating the HPG axis. And sort of there's a cascade of events. Those animals were quite young, and so we're also looking over the course of age to see if it will also be exacerbated with age.
Okay, we'll take last question. The oocytes appear to be smaller in the images that you showed with CP3 dysregulation and UCHO1 knockout. Was that phenotype you saw, and do you have an idea of why that may be?
So those representative images were not necessarily representative of all of the oocytes. I think, there is a range of sizes, and they certainly, these animals can ovulate, and when they do, their oocytes are of normal size. So they have the capacity to produce a normal sized oocyte. We have not done a systematic analysis of all the oocyte sizes to compare average size or within a certain stage average size, which we really should do. It's a good idea. So I don't have, in-depth thoughts on that, but this is an observation that has been made by us and others, and I think it's something we need to revisit for sure.
Okay, thank you so much, Dr. Greif. If you have any further questions, please feel free to direct them and reach out directly to both speakers. Before we wrap up, I'd like to congratulate both Dr. Lord and Dr. Greif again for the Rising Star Awards. I would also like to remind everyone to keep an eye out for the registration link for the SSR annual meeting, which will be coming out very soon. Thank you for joining us today, and we look forward to seeing you all next month, March 19th, for our third Rising Star webinar. Bye-bye.